In this video, we will continue our discussion about the CD19-CAR-NK-92 cytotoxicity effect, or the pathways that plays a role in this CD19-CAR-NK-92 cytotoxicity. In the previous video, we have discussed the cytotoxicity pathway role in the well-established cell lines, where we have found out that the perforin or granzyme-related pathways plays a significant role in this NK-92 based cytotoxicities compared to the FasL and the other pathways. Now they wanted to check these pathway roles against the primary B cells. So they did the similar experiment. Only thing is that they changed the target cells to primary B cells, and they did it only for this one effector to target ratio, that is 1:1. So compared to the wild type NK-92 cells, this reference CAR NK-92 cells shows high cytotoxicity, which is expected. Thereafter, they use this FasL knockout CAR NK-92 cells. Here the cytotoxicity reduced, and this reduction is statistically significant. However, when they use this, this indicates that the first one, the FasL knockouts reduction in the cytotoxicity, indicates that the FasL plays a role towards the cytotoxicity against the primary B cells. However, when they use the perforin knockout, the reductions in the cytotoxicity is statistically significant, but it is much more higher compared to the FasL knockout. It is the cytotoxicity reduced to 4%. And when they check it for the dual knockout, the reductions, they are able to see the similar cytotoxicity that they have seen in the Perforin 1 knockout, indicating that the Perforin 1 pathways, Perforin or the granzyme pathways play a significant role compared to the FasL knockout or the other pathways. These things they have quantified using this analysis of this cell lysis results in terms of this percentage of cytotoxicity efficacies. There they are able to find out that around 75% of this efficacies, or 75% of the cytotoxicities is based on this perforin or granzyme pathways, and the 18% is related to the FasL or FASR interactions, and the 7% is related to other pathways. So this from these results they come to a point that this NK-92 CAR cells cytotoxicity can be enhanced if they have the antigen informations that are expressed on the target or the tumor cells. So in this case, what antigen informations that they have used is the CD19 that are usually expressed on the primary B cells. So they used CD19 expressed CAR cells. So this results in this enhanced cytotoxicities that can be seen compared to the wild type NK-92 cells. The one thing that we need to know that even wild type NK-92 cells has baseline cytotoxicity against the target cells, and this could be because the NK-92 cells are already the immune cells, and they have the capability to detect the tumor cells or the cells which are infected with virus. And one of the receptors that plays a role towards this direction is GKD receptors. So these GKD receptors, I might be spelling it wrong, but these GKD receptors could easily detect the virus infected cells or the target tumor cells, and can act against them using the perforin or granzyme pathways. And the other things, that in the dual knockout, the decrease is not statistically significant compared to the perforin knockout, because the perforin knockout already contributes significantly, that is 75%. That is the other dual knockout could contribute the 18%, and those reductions might not reflected so much that the difference becomes statistically significant. So from these results, from the results that are presented in the previous figure, or the previous video, they made a point that the CD19 CAR-NK-92 cells can act based on the perforin granzyme pathways mainly, because it shows the similar high cytotoxicity contribution in the primary B cells and also in the well-established cell lines. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.